And welcome back to another chapter of Advanced Pathophysiology. Today we talk about Chapter 6, the epigenetics of disease. So jumping right in, humans exhibit an impressive diversity of physical and behavioral traits. Much of this diversity is attributable to genetic variation, another substantial contributor of epigenetic modification. The specific definition of epigenetic remains a topic of discussion among biologists. Under one definition, the term is reserved for modifications that are not encoded in nucleotide sequence, but are nevertheless transmitted when a somatic cell divides, when gametes are produced, or both. In this chapter, the term epigenetic is used to refer to the process that modulate how a given set of genomic information gives rise to a phenotype. Under this definition, epigenetic mechanisms include chemical modification to DNA and associated histones that produce a small regulatory RNA molecule, and more generally, gene regulation by epigenetic process at the level of either transcription or translation. Epigenetic modification is essential to fundamental processes of human development, including the differentiation of embryonic stem cells into specific cell types and the inactivation of one of the two X chromosomes in each cell of the genetic female. Some genes are said to be imprinted, meaning that they are inherited from the mother and the father in predictable and distinct epigenetic states. At imprinted loci, it is always either the allele from the mother or the allele from the father that is expressed in the offspring. A variety of diseases can result from abnormal epigenetic states. For example, metabolic disease can occur when there are abnormal expressions from both alleles at the locus that is typically imprinted. Exposure to environmental stressors can markedly increase the risk of abnormal epigenetic states and is strongly associated with some cancers. Because of the increased clear rule in a wide range of pathologies, such epigenetic perturbations are currently a focus of both preventative efforts and pharmaceutical intervention. So, our overview of epigenetic mechanisms. Epigenetic marks include DNA methylation and hydroxymethylation, chemical modifications that alter the charges of the histone proteins around which DNA is bound for compaction with the nucleus, and RNA-based mechanisms. Your DNA methylation. DNA methylation occurs through the attachment of the methyl group to the carbon 5 position of the cytosine. In some somatic cells of adults, DNA methylation occurs principally at cytosines that are followed by a genuine or guanine base, sometimes known as the cystosine and CpG dinucleotides. In human embryonic stem cells, methylation also can occur at cytosines outside of the CpG context. DNA methylation plays a prominent role in human health and disease. For example, in each cell of a normal human female, one of the two X chromosomes has dense methylation at most regions. Dense methylation is a key feature of heterochromatin, a structure consisting of DNA tightly wound around histones into a condensed state and not actively transcribed. The heterochromatization of the so-called inactive X accounts for the cytologic observation that each cell from a human female has a region of densely stained DNA. This region was later confirmed to be highly condensed inactive X chromosome. Most regions of the other X chromosome in a cell from a human female, the so-called active X chromosome, are largely devoid of DNA. Methylation. The active X chromosome is said to be transcriptionally active euchromatic state. Epigenetic inactivation of one of the two X chromosomes occurs in each cell of a human female during gastrulation, a phase of early embryonic development. The determination of which chromosome is to be silenced, either the copy from the father or the copy from the mother, occurs at random and independently in each cell. The silent state of that chromosome and the active state of the other are inherited by descendant cells. If a woman's two X chromosomes carry different alleles at a given locus, random X inactivation in early development can lead to somatic mosaicism, wherein differences between the alleles active in two cells can confer to very different traits. Visually notable examples include the patchy coloration of calico cats and anhydrotic ectodermal dysplasia, the patchy presence and absence of sweat glands in the human skin, females with one X chromosome bearing a normal allele and one X chromosome bearing a mutant allele. It is important to note that somatic mosaicism, because of random inactivation, can arise for any X-encoded trait. As a result, females who inherit one normal allele and one disease allele at an X-encoded gene tend to have less severe disease phenotypes than do males whose lone X chromosome bears a disease allele. This pattern is reflected in the typically lower severity in females as compared to males for color blindness, Fragile X syndrome, and other phenotypes that arise from mutations on the X chromosome. Aberrant DNA methylation, either the presence or dense methylation where it is typically absent or the absence of methylation where it is typically present, is associated with misregulation of tumor suppressor genes and oncogenes. Specific alterations to DNA methylation states are a common feature of several human cancers, including those of the colon, breast, and details of these alterations can be useful in assessing disease prognosis. Your DNA hydroxymethylation. DNA hydroxymethylation is the most common in cells that are undergoing epigenetic transition. DNA hydroxymethylation differs from DNA methylation in that it is a hydroxymethyl group, rather than methyl group, that is affixed to the C5 of cytosine. Discovery of this alternate DNA modification has helped to resolve long-standing uncertainties as to how genomic regions dense in DNA methylation undergo methylation loss, as observed, for example, in early embryonic development. In 2011, Wiedemann and colleagues found that genome-wide decline in DNA methylation in early murine zygotes occur in concert with genome-wide increase in DNA hydroxymethylation. They also found that this loss of methylation, as well as the parallel gain in hydroxymethylation, were obliterated in zygotes deficient in the TET3 enzyme. These two findings help to establish that TET enzymes can convert DNA methylation into hydroxymethylation. The name TET derived from the term 10-11 translocation, a reference to the discovery of these enzymes through their association with a chromosome translocation between chromosome 10 and 11 that is commonly observed in some forms of leukemia. Though the specific function impacts of hydroxymethylation remain an area of active investigation, one recent proposal is that it is associated with the activation of lineage-specific enhancers, and so can play a role in specifying cell lineages. Histone modification. Histones are the positively charged proteins around which negatively charged DNA molecules are wound, facilitating compaction of DNA into the cellular nuclei. When all the DNA that comprises the human genome is wound around histones, it is only about 1 out of 40,000 as long as it would be in its uncondensed state. A set of histones and the segment of DNA wound around them are together known as individual nucleosomes. The total chromatin in a cell, a combination of DNA and its associated histones and RNA molecules, is made up of individual nucleosome units. When a given segment of DNA is bound tightly, its associated histone, it is said to be heterochromatic, such that, as noted previously in describing the inactive X chromosome, it is not accessible by transcription factors, and so cannot be used to guide the production of mRNA. By contrast, if a given segment of DNA is only loosely bound to associated histones, it is said to be euchromatic, such that, as noted previously in the describing that active X chromosome, transcription factors are able to access it and use it as a template for their production of RNA. Whether a given segment of DNA is heterochromatic or euchromatic depends largely on chemical modification to histone tails, parts of the histone protein complex that extends away from the main part of the nucleosome. Researchers are only beginning to understand the full diversity and complexity of these histone modifications. The two modifications best studied are histone acetylation and histone methylation. Histone acetylation tends to diminish the positive charge of histones, reducing the strength of their binding to negatively charged DNA, and ultimately making DNA more accessible for transcription. Histone methylation can either increase or decrease the strength of bonding between DNA and histones, depending on the specific parts of the histone to which the methyl groups are added. Whether individual segments of the genome are heterochromatic or euchromatic, states play a critical role in determining the development potential of a given cell. That is, its capacity to give rise to a diverse set of differentiated cell types. For example, chromatin states differ substantially between embryonic stem cells, which are poised to give rise to all of the different cell types that comprise the individual, and terminally differentiated cells, which are committed to a specific developmental path. The fraction of DNA that is in the heterochromatic state typically increases as cells differentiate, in parallel to the reduction in the number of genes that are active as a cell lineage transition from pluripotency, when they have the capacity to give rise to a large number of descendant cell types, to terminal differentiation, and when they are able to produce only a cell of a single specific type. Mutations in genes that encode histone-modifying proteins have been implicated in various pathologic states, including congenital heart disease, highlighting histone modification as critical to normal development. In contrast to the vast majority of other cell types, including oocytes, sperm cells express not histones, but protamines, which are evolutionarily derived from histones. Protamines enable sperm DNA to achieve compaction even greater than for the histone-bound DNA in somatic cells. This tight compaction improves the hydrodynamic features of sperm cells, facilitating their movement. Your non-coding RNA. Non-coding RNAs play an important role in regulating a wide variety of cellular processes, including RNA splicing and DNA replication. Of particular relevance to gene regulation are microRNAs, which are encoded by DNA sequences approximately 22 nucleotides long and typically reside within the introns of genes or in non-coding intergenic regions. In contrast to DNA methylation and histone modification, both of which principally impact gene expression at a level of transcription, your microRNA typically modulates the stability and translation efficiency of messenger RNAs. Encoded at the loci, interaction between your microRNA and your messenger RNAs, the target for degradation is typically mediated by regions of partial sequence complementary. As a result, microRNA can at once be specific enough so that they do not bind to all of the messenger RNA in a cell and general enough to regulate a large set of different messenger RNA sequences. MicroRNA can also modulate translation by impairing ribosomal function. MicroRNA regulate diverse signaling pathways. Those that stimulate cancer development and progression are called oncomers. For example, microRNAs have been linked to carcinogenesis because they alter the activity of oncogenes and tumor suppressor genes. Like other classes of genomic elements, the sequence that encode microRNA can be transcriptionally silenced by DNA methylation, insofar as the expression of microRNA can modulate the formation and growth of tumors. Epigenetic modification of the sequence that encode them is a key area of exploration of strategies for characterizing and perhaps even treating cancer. Your epigenetics and human development. Each of the cells in the early embryo has the potential to give rise to a somatic cell of any type. These embryonic stem cells are therefore said to be totipotent, possessing all power. A key process in early development is the differential epigenetic modification of specific DNA nucleotide sequences in their embryonic stem cells, ultimately leading to the differential gene expression profiles that characterize differentiated somatic cell types. Early modification ensures that specific genes are expressed only in the cells and tissue types in which their gene products typically function. Epigenetic modifications early in development processes also highlight a fundamental feature of genetic as compared to epigenetic information. All of the cells in a given individual contain almost exactly the same genetic information. It is the epigenetic information eventually placed on top of these sequences that enables them to achieve the diverse functions of differentiated somatic cells. A small percentage of genes, termed housekeeping genes, are necessary for the function and maintenance of all cells. These genes escape epigenetic silencing and remain transcriptionally active in all or nearly all cells. Your housekeeping genes include those encoding histones, DNA and RNA polymerases, and ribosomal RNA genes. So, how do embryonic stem cells achieve epigenetic states typically of total potency, whereby they can give rise to all of the diverse cell types that comprise a fully developed organism? One explanation is that early embryogenesis, which occurs during the 10 or so days just after fertilization, is characterized by rapid fluctuation in genome-wide DNA methylation densities. Fertilization triggers a global loss of DNA methylation at most loci in both the oocyte-contributed and the sperm-contributed genomes. This loss of methylation is accomplished in part by suppression of the DNA methyltransferases, the enzymes that add methyl groups to DNA. Methylation is not directly copied by the DNA replication process. Instead, immediately following the replication, the methyltransferases read the pattern of methylation on the parent DNA strand and use that information to determine which daughter strand cytosines should be methylated. As embryonic cell division continues, eventually yielding cells that have nearly all of their loci in unmethylated transcriptionally active states, around the time of implantation in the uterus, the DNA methyltransferases become active again, permitting establishment of the cell lineage, specific marks required for the establishment of organ systems. So your genomic imprinting. A baby inherits two copies of each autosomal gene, one from its mother and one from its father. For a large subset of these genes, expression is biallelic, meaning that both the maternal and the paternal inherited copies contribute to offspring phenotypes. As noted earlier, for another smaller subset of these genes, expression is stochastically monoallelic, meaning that the maternal copy is randomly chosen for inactivation in some somatic cells, and the paternal copy is randomly chosen for inactivation in other somatic cells. For a third and smaller subset of autosomes, either the maternal copy or the paternal copy is imprinted, meaning that either the copy inherited through the sperm or the copy inherited through the egg is inactivated and remains in this inactive state in all of the somatic cells of the individual. The subset of genes that are subject to imprinting is highly enriched for loci relevant for organismal growth. The genetic conflict hypothesis was developed as a potential explanation for this pattern. The logic of this hypothesis is as follows. Although both the mother and the father benefit genetically from the birth and the survival of offspring, their interests are not entirely aligned. Because mothers make a large physiologic investment in each child, it is in their evolutionary best interest to limit the flow of energetic resources to any given offspring, so as to maintain their physiologic capacity to bear subsequent children. By contrast, except in cases of lifelong monogamy, it is in the best interest of the father for the child to extract maximum resources from its mother, since the father's reproductive success is tied to the survival of their own child, but not to the mother's ability to bear additional offspring in the future. Therefore, imprinted genes expressed from the maternal genome are predicted to limit offspring size, whereas imprinted genes expressed from the paternal genome are predicted to result in larger offspring. Available data are broadly consistent with these predictions. One hallmark of imprinting-associated disease is that phenotype of affected individuals is critically dependent on whether the mutation is inherited from the mother or from the father. Some examples are included in the following. Your Prader-Willi and Angelman syndromes. A well-known disease example of imprinting is associated with the deletion of about 4 million base pairs of long arm of chromosome 15. When this deletion is inherited from the father, the child manifests Prader-Willi syndrome, with features that include short stature, hypotonia, small hands and feet, obesity, mild to moderate intellectual disability, and hypogonadism. The same deletion, when inherited from the mother, causes Angelman syndrome, which is characterized by severe intellectual disability, seizure, and ataxic gait. These diseases are each observed in about 1 of 15,000 live births. Chromosome deletion are responsible for about 70% of cases of both diseases. The deletion that cause Prader-Willi and Angelman syndrome are indistinguishable at the DNA sequence level and affect the same group of genes. It was unclear for several decades how the same deletion could produce such disparate results in different individuals. Further analysis showed that the 4 million base pair deletion, the critical region, contains several genes that are normally transcribed only on the copy of the chromosome 15 that is inherited from the father. These genes are imprinted on the copy of chromosome 15 inherited from the mother, meaning that they are transcriptionally silenced. Similarly, other genes in the critical region are transcriptionally active, only on the chromosome copy, inherited from the mother, and are inactive, or imprinted, on the chromosome inherited from the father. If the single active copy of one of these genes is lost because of a chromosome deletion, then no gene production is produced, resulting in disease. Molecular analysis has revealed a great deal about genes in this critical region of chromosome 15. The gene responsible for Angelman syndrome encodes a ligase involved in protein degradation during brain development, an observation that may help to explain the intellectual disability and ataxia observed in this disorder. In the brain, this gene is active only on the chromosome copy inherited from the mother. Consequently, a maternally transmitted deletion removes the single active copy of this gene. Several genes in this critical region are associated with Prader-Willi syndrome, and they are transcribed only on the chromosome transmitted by the father. A paternally transmitted deletion removes the only active copies of these genes, producing the features of Prader-Willi syndrome. So, Beckwith-Wiedemann syndrome. Another well-known example of imprinting is Beckwith-Wiedemann syndrome, an overgrowth condition accompanied by an elevated risk of cancer. Beckwith-Wiedemann syndrome is usually identifiable at birth because of the large size of gestational age, neonatal hypoglycemia, a large tongue, creases on the earlobe, and omphalocele, a birth defect in infant intestines. Children with Beckman-Wiedemann syndrome have an increased risk of developing Wilms tumor or hepatoblastoma. Both of these tumor types can be treated effectively if they are detected early. Thus, screening at regular intervals is an important part of management. Some children with Beckwith-Wiedemann syndrome also develop asymmetrical overgrowth of a limb or one side of the face or trunk, your hemihyperplasia. As with Angelman syndrome, a minority of Beckwith-Wiedemann syndrome cases are caused by the inheritance of 2 copies of chromosome 11 from the father and no copy of chromosome from the mother, in a process known as uniparental disharmony. Several genes on the short arm of chromosome 11 are imprinted on either the paternally or maternally transmitted chromosome. These genes are found in 2 separate differentially methylated regions, DMRs. So, in And DMR1, the gene that encodes insulin-like growth factor 2, is inactive on the maternally transmitted chromosome but active on the paternally transmitted chromosome, such that a normal individual has only one active copy of IGF2. When two copies of the paternal chromosome are inherited, or there is a loss of imprinting on the maternal copy of IGF2, an active IGF2 gene is present in double dose. These changes produce increased levels of IGF2 during fetal development, contributing to the overgrowth features of the Beckwith-Wiedemann syndrome. Thus, in contrast to Prader-Willi and Angelman syndrome, which are produced by a missing gene product, Beckwith-Wiedemann syndrome is caused in part by the overexpression of gene product. And then the Russel-Silver syndrome. Russel-Silver syndrome is characterized by growth retardation, proportionate short stature, leg length discrepancy, and a small triangular-shaped face. About one-third of Russel-Silver syndrome cases are caused by imprinting abnormalities of chromosome 11p15.5 that lead to downregulation of IGF2 and therefore diminishes growth. Another 10% of cases of Russel-Silver syndrome are caused by maternal uniparental disomy. Thus, while it is upregulation or extra copies of active IGF2 that causes overgrowth in Beckwith-Wiedemann syndrome, it is downregulation of IGF2 that causes diminished growth, seen in Russel-Silver syndrome. Your epigenetics in cognitive development and mental health. Several lines of evidence suggest a role of epigenetic abnormalities in disorders of cognitive development. Your epigenetic and ethanol exposure in utero. The impact of ethanol exposure in utero on skeletal and neural development was first reported in 1973 and led to broad awareness of fetal alcohol syndrome. It was not until recently, however, that population-based and molecular level studies began to clarify the epigenetic signal that are associated with the development abnormalities. Initially, researchers found that alcohol exposure in utero can impact the DNA methylation states of various genomic elements, but without specific emphasis on loci directly relevant to skeletal and neural development. Later experiments revealed that treating cultured neural stem cells with ethanol impairs their ability to differentiate to functional neurons. This impairment seems to be correlated with aberrant dense methylation at loci that are active in normal neuronal tissue. One possible explanation for these effects is the ethanol exposure in utero modulates fetal expression of the DNA methyltransferases. Your epigenetics and mental health. Several emerging lines of evidence suggest an association between altered epigenetic states and mental health. A recent study found that children who grew up under conditions of poverty have atypical methylation at a serotonin level receptor, suggesting DNA methylation as a mechanistic link between early life socioeconomic stress to an increased propensity to depression. Similarly, in individuals with post-traumatic stress disorder, alterations in gene expression in key neural pathways are associated with atypical methylation in a large set of genes. Among individuals with PTSD, specific changes in methylation are associated with suicidal ideation and behavior. Autism spectrum disorder also is associated with altered DNA methylation at some loci. In some cases, as in the connection of epigenetic abnormality to depression and PTSD, it is not clear whether epigenetic alteration plays a mechanistic role in the etiology of cognitive syndromes, or whether information on this link is useful principally for suggesting biomarkers potentially useful for diagnosis. Fragile X syndrome, a genetic epigenetic syndrome. Fragile X syndrome can be described as a genetic and epigenetic disease, insofar as abnormalities of both sorts are observed at the Fragile X locus, FMR1, in affected individuals. The most common genetic abnormality at FMR1 involves expansion in a number of cytosine guanine dinucleotide repeats in the same gene encoder or promoter. Females who have a cytosine guanine repeats in excess of the plus 35 that are typical at this locus are at risk for Fragile X-associated primary ovarian insufficiency, characterized by an elevated risk of early menopause. Males with moderate expansions are at risk of Fragile X tremor ataxia syndrome, characterized by a late onset intention tremor. Both of these conditions seem to arise through accumulation of excess levels of FMR1 mRNAs in nuclear inclusion bodies. Individuals with 200 repeats are at risk of Fragile X syndrome, characterized by reduced IQ and a set of behavioral abnormalities. Remarkably, while possession of a large CG repeat and FMR1 promoter dramatically increase the probability that an individual will have Fragile X syndrome, the disease can be at once present in males who have the large repeat and absent in their brothers who have inherited an allele of a very similar size. This can be explained at least in part by the observation that acquisition of methylation-based silencing at FMR1 is stochastic, meaning that the presence of large repeat increases the probability of the dense promoter methylation that could lead to gene silencing, but does not guarantee it. It remains to be seen whether dietary or environmental features can modulate the probability that dense methylation at FMR1 will accrue in individuals with full mutation alleles. Exploring the possible multigenerational persistence of epigenetic states that arise in response to environmental factors. Emerging data suggests that conditions encountered in utero during childhood and even during adolescence or later can have long-term impacts on epigenetic states. Some data have been interpreted to indicate that epigenetic changes arising during an individual's lifetime can be transmitted through the germline, with the potential to impact gene expression in the future. Some reports of transgenerational inheritance of environment-induced epigenetics have failed to be replicated in subsequent work by other researchers. For example, Iqbal and colleagues sought to follow up on an earlier report that the epigenetic impacts of endocrine disruptors can be transmitted by an unknown mechanism to the germline of the grandchildren of the mice exposed. Although they did confirm the capacity of endocrine disruptors to induce epigenetic changes in tissue and gametes directly exposed, they found no evidence that any differences can persist to the germline of exposed animal grandchildren. Further investigation will be required to explain the disparities between these conflicting findings. A few examples of such apparent connections between environment and inheritance of epigenetic states are discussed below. All of the examples remain under active investigation, however. So, epigenetic and nutrition. During the winter of 1943, millions of people in urban areas of the Netherlands suffered starvation conditions as a result of a Nazi blockade that prevented shipments of food from agricultural areas. When researchers sought to investigate how exposure to famine during the Dutch hunger winter had impacted individuals born in a historically prosperous country rather, they found that individuals who had been in utero during the interval of severe nutrition deprivation were more likely to suffer from obesity and diabetes as adults than were the other individuals from the Netherlands. There also seemed to be a transgenerational impact in that the children of individuals who were in utero during the Dutch hunger winter were found to be significantly smaller than were the children of those not impacted by the blockade. Other datasets reveal elevated risk of cardiovascular and metabolic disease for offspring of individuals exposed during early development to fluctuations in food availability. The specific molecular mechanism that may mediate these apparent relationships between nutritional deprivation and disease risk on one or more generation are largely unknown. From some animal models, it seems that the IGF2 gene is a possible target of epigenetic modifications, arising through nutritional deprivation, exposure in utero, and through lactation to some chemicals. Seems to be the lead to epigenetic modifications similar to those that arise through nutritional deprivation in early life. Your epigenetic and maternal care. There's increased evidence that parenting style can impact epigenetic states and that this information can be transmitted from one generation to the next. Mice of the other rodents can exhibit two alternate styles of nursing behavior. One style is characterized by frequent arched back nursing, with a high level of licking and grooming behavior. An alternate style is characterized by infrequent arched back nursing and less licking and grooming behavior. In one study, pups of mothers that engaged in frequent arched back nursing were found to have significantly lower methylation levels and higher transcription activity of a glucocorticoid receptor encoding locus. Because the glucocorticoid receptor is involved in a pathway that intensifies fearfulness and response to stress, these findings suggest that alteration to methylation states could help maintain the finding and exposure to stress early in life and can modulate behavior in adulthood. These findings also highlight the concept that epigenetic processes can help store information about the environment and that the relevant epigenetic modifications can modulate behavior later in life. So, epigenetic change over the lifespan. Twin studies reveal epigenetic changes over time. Identical monozygotic twins whose DNA sequences are essentially the same offer a unique opportunity to isolate and examine the trajectory of epigenetic change over the lifespan. A recent study found that as twins age, they exhibit increasingly substantial differences in methylation patterns of the DNA sequences of their somatic cells. These changes are often reflected in increasing numbers of phenotypic differences. Twins with significant lifestyle differences tend to accumulate larger number of differences in their methylation patterns. These results, along with findings generated in animal studies, suggest that changes in epigenetic states may be an important part of the aging process. So, epigenetics and aging. The occurrence of epigenetic change over the lifespan, as suggested by studies in twins, is echoed by broader studies in both human and non-human systems. In yeast, a shift in histone modification, as well as an overall decline in histone abundance, is a marker of replicative aging and is associated with dysregulation of gene expression. Chemical labeling techniques have revealed that the genome-wide abundance of hydroxymethylcysteine increases with time in the brain of adult mice. In view of these and similar findings, some authors have proposed that senescence itself can be characterized as an epigenetic phenomenon. From this perspective, while accrual of adverse exposure over the lifespan, it is to some degree unavoidable. Understanding environmental impacts on epigenetic states could lead to a new paradigm for lifespan extension. This apparent relationship between epigenetics and aging could help us explain the long-standing observation that metformin, a drug used to treat insulin resistance, also is effective in slowing senescence in yeast. Studies in human populations suggest that long-term use of metformin may even extend mean lifespan in diabetic people beyond the expected for untreated, non-diabetic individuals. Initially, these effects, which echo the increase in lifespan occurring under caloric restriction in primate models, were thought to operate directly through the impact of metformin on metabolism. However, metformin has recently been found specifically to alter the expression of SIRT1, the gene encoding sirtuin, a protein that modulates the metabolic response to energy limitation. Therefore, it is possible that the drug that has been used to treat insulin resistance may modulate epigenetic pathways, with possible opportunities for lifespan extension in humans. So, molecular and computational tools for exploring epigenetic states. Epigenetic information is not encoded DNA sequence. Therefore, specialized methods must be used to detect and quantify it. So detecting DNA methylation in populations of molecules, a methyl-sensitive restriction enzyme. Many restriction enzymes are sensitive to methylation, and so they cleave DNA only at unmethylated cytosines. Clues to the overall level of methylation in DNA isolated from a given individual can therefore be gathered by treating DNA with a methyl-sensitive enzyme, and then comparing banding patterns for enzyme-treated and non-enzyme-treated samples. Detecting DNA methylation in a single molecule, via bisulfite conversion. Sometimes it is of interest to gather information about methylation on individual molecules. For example, among males who inherit a full mutation Fragile X allele, there is a strong correlation between the severity of the syndrome and the number of cells in which the Fragile X allele is densely methylated. To collect information on the methylation states of individual molecules, DNA can be subjected to bisulfite conversion before sequencing. Bisulfite treatment does not alter most nucleotides, including methylated cytosines, but deaminates unmethylated cytosines to uracil. Because uracil complements adenine, not guanine, in double-stranded DNA, methylated and unmethylated cytosines can be distinguished in conventional sequence data, providing that the original sequence of that region is known and can be used to distinguish between true thymines and converted, unmethylated cytosines. Either subcloning followed by Sanger sequencing or single molecule sequencing technologies can then be applied to ascertain the detailed methylation patterns of individual molecules. So, detecting DNA hydroxymethylation, your fluorescence resonance energy transfer. Following Wiedemann and colleagues discovered that loss of DNA methylation and the gain of DNA hydroxymethylation during early development, there has been considerable interest in exploring the specific functional impacts and mechanisms of this transition. However, efforts in this area have been hampered by lack of method to detect relative levels of DNA methylation and DNA hydroxymethylation within individual molecules. Recently, Song and colleagues introduced a promising solution. Under their new method, DNA are first treated with different fluorophores. Of these, one binds methyl groups and the other binds hydroxymethyl groups. When samples are examined using fluorescence assays, the methylation and hydroxymethylation can be distinguished visually. This new method will undoubtedly enable critical insight into the basic biology of epigenetic transition and perhaps into the mechanisms of epigenetic disease. Identifying histone modification states, your chromatin immunoprecipitation. The identity of DNA-bound proteins can provide important clues as to the activity states of individual regions. The goal of chromatin immunoprecipitation is to ascertain the sequence bound by specific proteins of interest. Broadly, this is achieved in 7 steps. The cross-link, the shear, select, capture, remove, sequence, and repeat. I'll leave that up for just a moment so you can look at what each of those steps individually do, and we'll keep going. Detecting DNA accessibility states. DNA's hypersensitivity testing. DNA's enzymes cleave uncondensed, transcriptionally active euchromatic DNA, but not condensed, transcriptionally silent heterochromatic DNA. Genomic regions that are euchromatic in a given cell type therefore can be identified through DNA's treatment of bulk DNA samples. This approach can be readily applied at the genome scale, opening opportunities to compare transcriptionally active states across cells under various differentiation states and culture conditions. Detecting DNA accessibility states in single molecules. Assay for transposase accessibility chromatin. Like DNase hypersensitivity testing, the recently introduced assay for transposase accessible chromatin, your ATAC-Seq, is focused on identifying genomic regions whose open euchromatic states suggest that they could be transcriptionally active. However, contrast to DNase assays under which transcriptionally active states are identified by their overall sensitivity to DNase enzyme cleavage, ATAC sequencing uses a transpose, for which cleavage of accessible sites is followed immediately by specific ligation of a linker that bears barcodes that differ among cells. Genomic regions that have been ligated to adapters can then be isolated, amplified, and sequenced, in contrast to the earlier approaches, which collect population-level summaries of active and inactive states of individual genomic locations. The use of single-cell barcodes and ATAC sequencing enables comparison of epigenomes across individual cells in the sample. Using single-cell ATAC sequencing, one group found that there is an unexpected high level of variation among the epigenetic states of individual intestinal lymphoid cells, and that this variation is shaped by the microbiome. Publicly available resources enable exploration of epigenetic states at genome scale. One of the great advances of genomic era is the emergence of public online data repositories for researchers to share their findings, enable insight that might not be accessible from the work of any single research group. Such publicly available datasets have provided unprecedented opportunities to compare epigenomic profiles over developmental time and between tissues in disparate functional states. The Encyclopedia of DNA Elements, a project supported by the National Human Genome Research Institute, is providing great insight as a catalog of the epigenetic signatures of cell growth and culture. The Roadmap Epigenomics project, a cooperative venture of epigenomic biologists worldwide, sponsored by the U.S. National Institute of Health, is expanding this goal to provide information on freshly sampled cells and tissues. Although it is possible for users to browse data using a visual interface, the most efficient way to leverage these publicly available resources is to use a command line approach to download, process, and analyze available datasets. In just the past 3 years, data available through Roadmap have provided insight into the genomic architecture underlying disorder of gonadal development, juvenile arthritis, and attention deficit hyperactivity disorder. Your epigenetics and cancer. DNA methylation and cancer. Some of the most extensive evidence for the association of epigenetic modification with human disease comes from studies of cancer. Tumor cells often exhibit decreased methylation genome-wide relative to normal cells of the same type, which can increase the activity of oncogenes. Genome-wide loss of methylation tends to continue as tumors progress from benign neoplasms to malignancy. In addition, the promoter region of tumor suppressor genes are often hypermethylated, decreasing their rate of transcription and thus their ability to inhibit tumor formation. Hypermethylation of the promoter region of the RB1 gene is often seen in retinoblastoma, and hypermethylation of the MLH1 locus is associated with colorectal cancer. It is important to note that the observation of perturbed DNA methylation states in tumors is not itself an indication that perturbation in DNA methylation state is the proximate cause of cancer. However, as described in the following, the findings that many tumors have epigenetic abnormalities hint at existing new opportunities for minimally invasive cancer screening diagnosis. So here we're looking at the comparing molecular mechanisms of Fragile X and FSHD. So I'll leave that up for just a moment, and then we'll keep going. Global epigenomic alterations and cancer. A major feature of one form of inherited colon cancer is the methylation of the promoter region of the gene, MLH1, whose protein product repairs damaged DNA. Inactivation of MLH1, a DNA mismatch repair enzyme, is associated with accrual of DNA damage. Another common feature is colon tumors. Abnormal methylation of tumor suppressor genes also is common in the progression of Barrett's esophagus, a condition in which the esophagus is lined with cells that have features typically associated with the lower intestinal tract. Epigenetic screening for cancer. As noted previously, specific epigenetic abnormalities are often defining features of a tumor, but cannot necessarily be inferred to the cause of disease, regardless of the nature of the connection between epigenetic abnormality and any particular cancer. The ability to screen for abnormal epigenetic states raises the possibility that epigenetic screening approaches could complement or even replace existing early detection methods. In some cases, epigenetic screening can be implemented using bodily fluids, such as urine or sputum, eliminating the need for more invasive, costly, and risky strategies that are currently standard. For example, screening for epigenetic misregulation in microRNAs has shown promise as a tool for detecting and characterizing cancers of the colon, breast, and prostate. Other epigenetic-based screening approaches have shown promise for detecting and characterizing cancers of the bladder, lung, and prostate. Screening for epigenetic abnormality to detect cancer may be especially powerful in conjunction with emerging methods for detecting tumors in blood and sputum samples. These liquid biopsies methods are minimally invasive and so could be useful not only for characterizing abnormalities after diagnosis, but also for performing routine, simultaneously screening for multiple types of cancers, even in individuals without signs of disease, and for detecting the emergence of drug resistance mutations in individuals undergoing chemotherapy. Your misregulation of microRNAs in cancer. Your hypermethylation also is seen in microRNA genes, which encode small RNA molecules that bind to the end of mRNAs, degrading them and preventing them translation. More than 1,000 microRNA sequences have been identified in humans, and hypermethylation of specific subgroups of microRNA is associated with tumorigenesis. Emerging strategies for treatment of epigenetic disease. Epigenetic modifications are potentially reversible. DNA can be demethylated. Histones can be modified to change the transcriptional state of nearby DNA, and microRNA encoding loci can be up or down regulated. This raises the prospect for treating epigenetic disease with pharmaceutical agents that directly reverse the changes associated with the disease phenotype. In recent years, intervention involving all three types of epigenetic modulators have shown considerable promise for the treatment of disease. So DNA demethylating agents, your 5-azacitidine, has been used as a therapeutic drug in the treatment of leukemia and myelodysplastic syndrome. A cytosine analog, 5-azacitidine, is incorporated into DNA opposite its complementary nucleotide guanine. Chemical differences between 5-azacitidine and conventional cytosine cause it to achieve irreversible binding to the DNA methyltransferases. As a result, the administration of the medication tends to reduce the density of DNA methylation, offering prospects for reversing the aberrant accumulation of methylation at tumor suppressor loci. Though associated with various side effects, including digestive disturbances, treatment with the medication has shown promise in the treatment of diseases, including pancreatic cancer and myelodysplastic syndromes. Here's a picture of that medicine. And then histone deacetylase inhibitors. To the activity of the histone deacetylases increases chromatin compaction, decreasing transcriptional activity. In many cases, excessive activity of histone deacetylases results in transcriptional inactivation of tumor suppressor genes, leading ultimately to the development of tumors. Treatment with this medication inhibitors, either alone or in combination with other drugs, has shown promise in reducing cell division rates in culture cells of cancer of the breast, prostate, and pancreas. Most recently, this medication has been used to treat some forms of leukemia and lymphoma that are characterized by transcriptional repression. In those diseases, this drug becomes overly active, removing the acetyl groups that diminish the strength of bonds between DNA and histones, ultimately preventing transcription factors from accessing DNA regions whose activity is required for proper cellular function. A large number of drugs have proven useful in diminishing the removal of acetylation from histones. One drug in particular, eboxinostat, is undergoing safety assessment to follow up on encouraging outcomes in a phase 2 clinical trial. Your microRNA targeting. A major challenge in developing drugs that modify epigenetic alterations is to target one of the genes responsible for specific cancer. Therapeutic approaches that use microRNAs offer a potential solution to this problem because treatment can be targeted to individual loci that have aberrant expression using sequence characteristics of relevant RNA molecules. So what are your future directions? Robust experimental observations are clarifying the role of epigenetic states in determining cell fate and disease phenotypes. The well-documented involvement of epigenetic abnormalities in carcinogenesis and the mounting evidence of the epigenetic changes in the other common diseases will likely elucidate possibilities for reversing the epigenetic abnormalities and possibly preventing their establishment in utero. And that concludes this chapter. We'll see you with chapter 22.